Start the class, madam. Start the class.
రాజయ్య వాడు రాత్రి
మేడం స్టార్ట్ చేయండి.
హలో మేడం, ఏమైంది?
Good morning, students. This is Rama JL SARVAIL. Yesterday discussed about the chemoosmosis, with which light phase or light reaction was over. And today we are going to discuss dark phase or dark reaction. This dark reaction is light independent reaction. That means it can take place in presence of light as well as in absence of light. And here, during light phase, ATP and NADPH are synthesized, and where they are used. Here, ATP and NADPH are used to drive the process leading to the synthesis of more accurate sugars And this dark phase is a biosynthetic phase of photosynthesis. And and this process, it is just now we said, it is not a light dependent. And end products of this light phase, ATP and NADPH, are utilized during the dark phase. Then this reaction, dark phase, is also called as biosynthetic phase or biosynthetic part. The reason for this is using of ATP and the reducing power, which are synthesized during the light phase. That's why it is called as biosynthetic phase pathway, and sugar synthesis will take place. And this process was explained by Melvin Kelvin by using C14, which is a radioactive carbon, for his experiments. And Kelvin used green algae exposed to the sunlight, and took some green algal cells and killed them in hot methanol. And after a few minutes, he examined the products which were obtained from this green algae, and to trace by means to trace this, that is why this kept tracing the path of the C14. Whichever compounds had the C14, he was able to interpret that these compounds, whatever by this they got entire cycle. Okay, the next and this Melvin this dark phase, Melvin Kelvin explained the dark reaction by using C14 carbon, and he killed green algae using hot methanol and examined products in the cells, and he found was a carbon compound called the phosphoglyceric acid, PGA, and this was identified as RUBP, and RUBP is a ketose sugar. The next equation of this is 6CO2 plus 12H2O gives rise to 6H2O6 plus 6H2O and 6O2 by absorbing sunlight in the presence of chlorophyll. This is the equation of photosynthesis. And actually entering into this Kelvin cycle. And this Kelvin cycle, Kelvin cycle, CO2 is first, it is accepted by a 2C compound. That's why 3C was formed, and anticipation to this is later on compounds which accept CO2 was treated as a 5C compound, and this was RUBP, and which is a ketone sugar and 5C compound. So here important thing is when CO2 is accepted by RUBP, that process is known as carboxylation. And in this, during this helping enzyme is rubisco, means ribulose carboxylase oxygenase. And this enzyme, ribulose carboxylase, it acts as sometimes it acts as a carboxylase and sometimes it acts as oxygenase. That means, depending upon the situation, it plays the role of carboxylase or oxygenase. The reason for that is this enzyme nature changes according to the factors like carbon dioxide, oxygen, and temperature. So, based on these factors, that rubisco acts as a carboxylase or oxygenase. The next, if we go through the cycle, this cycle is completed in 3 steps, or it is described in 3 stages. One is carboxylation, second is reduction, and third is regeneration of RUB. Here, briefly, the skeleton of Kelvin cycle. Here, carbon dioxide is absorbed by RUBP. Here it is RUBP. It combines with the CO2 and results in the formation of PGA. So this is first carboxylation. That means accepting carbon dioxide by RUBP is and resulting in the formation of PGA is a carboxylation. And in stage 2, these acids are converted into aldehydes. That is a second stage reduction. And in third stage, from aldehydes, partially few molecules are utilized in the preparation of sugar, and a major portion of our aldehyde molecules helps in regenerating RUBP. That is the third step, regeneration of RUBP. In this Kelvin cycle, these are the 3 main important steps, and the skeletal form is this. The next, first step is carboxylation. What is carboxylation? Carbon atom of a CO2 is fixed by RUBP and results in the formation of a 6-carbon compound, which is unstable, is formed, and it is PGA. RUBP plus CO2 gives rise to PGA. RUBP plus CO2 gives PGA, phosphoglyceric acid. And this reaction is catalyzed by the enzyme RUBP carboxylase. That means rubisco helps in this reaction. See here in cycle, carbon dioxide combines with RUBP. Carbon dioxide combines with RUBP in presence of rubisco and helps in the formation of PGA, phosphoglyceric acid. And this phosphoglyceric acid is a 3C compound, and 6 molecules, see here, 6 molecules are synthesized here, and so total number of carbons here 6 into 3, 18. So, and here this PGA undergoes phosphorylation and gives, and here Here, phosphoglyceric acid undergoes phosphorylation and results in the formation of 1,3-diphosphoglyceric acid. This 1,3-diphosphoglyceric acid that is also a 3C compound, and 6 molecules are synthesized. So here also 18 carbons are carried forward, and the next this 1,3-diphosphoglyceric acid undergoes reduction stage, that is the second stage. Reduction is the second stage of a Kelvin cycle. So during this second stage, during the reduction stage, acids are converted into aldehydes. Acids are converted into aldehydes. So that is the reduction stage. And here 1,3-diphosphoglyceric acid is reduced, or that undergoes reduction changes. And during this change, or for this change, it requires one agent, that is reducing agent, and this reducing agent or power which was synthesized during, just recall the light phase, that was synthesized during the light phase in the form of NADPH2, and this NADPH2 reduces, this NADPH2 reduces this 1,3-diphosphoglyceric acid into aldehydes. So is this clear? 1,3-diphosphoglyceric acid, which is a 3C compound, and forms synthesized 6 molecules. That means 6 into 3, 18C. That means tracking is same. 18 carbons. This 18 carbons, 1,3-diphosphoglyceric acid undergoes reduction, and for this reduction, it requires reducing power, which was synthesized during the light phase. Okay, by the help, by the support of this reducing power, 1,3-diphosphoglyceric acid molecules reduced into phosphoglyceraldehyde molecules by utilizing NADPH2, and that NADPH2 reduced into NADP. Here, molecules, how many molecules? Means 1,3-diphosphoglyceric acid molecules are 6, and totally they are reduced into 6 phosphoglyceraldehyde molecules. So each phosphoglyceraldehyde molecule requires 1 molecule of NADP. That's why total 6 NADPH2 are reduced into 6 NADP plus 6 IP, inorganic phosphate molecules. And now this phosphoglyceraldehyde is also a 3C compound, and synthesized molecules are 6. So totally 18. It's also a 18C compound. Out of 6 molecules, here out of 6 molecules, 1 molecule out of 6, 1 molecule goes to, or 1 molecule helps in preparing sugar or glucose, and the remaining 5 molecules, remaining 5 phosphoglyceraldehyde molecules, they are diverted to another way. So here, out of 6 PGAL, 1 PGAL is diverted to synthesize glucose. This is clear. Is this clear? Okay. The next, remaining 5 phosphoglyceraldehyde molecules. If you observe them, here, each here 5 IP out of this 5 phosphoglyceraldehyde molecules, two inorganic phosphate groups are separated, and remaining 3 phosphoglyceraldehyde molecules are utilized for the regeneration of RUBP. Here, phosphoglyceraldehyde, how many? The molecules are 5 molecules of phosphoglyceraldehyde, and which is a 3C compound. That means total number of carbons are 15. That means 5 into 3, 15 carbon molecules are there in PGAL. And out of this 5 PGAL, 2 phosphate IP inorganic phosphate groups are separated, and the remaining three are converted into ribulose 5-phosphate or ribulose monophosphate, in which the phosphate radical is connected with the 5th carbon. Here, so it is a 5C compound. Ribulose 5-phosphate is a 5C compound, and the synthesized molecules are 3. That means total number of carbons are 3 into 5, 15C. That means ribulose 5-phosphate, which is a 5C compound, are synthesized 3 molecules, and this each ribulose 5-phosphate molecule is carrying 1 IP, isn't it? So 3 molecules of ribulose 5-phosphate, it furtherly undergoes phosphorylation. Here, see, here that is this ribulose 5-phosphate or ribulose monophosphate that undergoes phosphorylation, and for this phosphorylation it requires 3 ATP molecules, and these 3 ATP molecules, they are utilized. That means by releasing ATP is will be utilized for this phosphorylation, and IP groups, inorganic phosphate radicals, they are connected with the ribulose monophosphates. See here, ribulose monophosphate, it is having only 1 IP. That means on 5th carbon is connected with the inorganic phosphate. So here 3 molecules of ribulose 5-phosphate, 3 molecules of ribulose monophosphate should convert into ribulose biphosphate. That is 3 molecules of ribulose biphosphate. That means for each ribulose 5-phosphate, 1 more IP should be added. For that sake, 3 molecules of ribulose, by utilizing 3 ATP molecules, turns into ribulose biphosphate, and this ribulose biphosphate again it gets ready to receive carbon dioxide molecule for carboxylation. That means to continue the Kelvin cycle, it is ready to receive carbon dioxide. So here the important thing is 6 molecules of phosphoglyceraldehyde, which is a 3C carbon compound. Here it is important. This phosphoglyceraldehyde, which is a 3C compound, it is synthesized. 6 molecules are synthesized. Out of 6 molecules, only one molecule is diverted to synthesize glucose. Remaining major molecules, remaining 5 molecules are diverted to regenerate RUBP. In this process, this PGAL, 5 molecules of PGAL, which is a 3C carbon compound. So 15 carbons are situated in this, And out of this 5, 2 phosphates, 2 molecules are diverted, and remaining 3 are converted into ribulose 5-phosphate or ribulose monophosphate. This is most important thing. So here, this monophosphate, each monophosphate molecule, there are 3 molecules, and each molecule is having 1 phosphate radical. That's why it is monophosphate, and that should become biphosphate. For that sake, it accepts or it utilizes 3 ATP molecules and converting ATP into ADP, and IP groups are added to this ribulose biphosphate, and that becomes RUBP, in which this monophosphate is also having 15 carbon molecules and RUBP also having the same number of carbon molecules. So that means the tracking is followed one after the other by maintaining the carbon molecules. Here, actually, here in the starting, we took 3 carbon dioxide molecules combined with the 3 RUBP. It is for understanding sake it was taken, but originally in nature, plants uses only 1 molecule of carbon dioxide. So if 1 molecule takes part in this Kelvin cycle, and here during the formation of PGAL, the molecule is that is divided into fractions. It is tough to understand in fractions. That's why, for understanding sake, here they took 3 carbon dioxide molecules and combined with the 3 RUBP, which facilitates to understand the cycle easily. Then this cycle is also called as C3 cycle. Why? Because the first formed compound, that means CO2, when CO2 is accepted by RUBP in presence of Rubisco, it synthesizes phosphoglyceric acid, which is a 3C compound. And this, because of the formation of this cycle is also called as a C3 cycle. Then here, see the points. Reduction. Second part of the carbon Kelvin cycle is carbon is a reduction of PGA to phosphoglyceraldehyde, PGAL, and this part includes the following reactions. That is, PGA is phosphoglycerated by phosphoglycokinase to yield biphosphoglyceric acid. ATP donates a phosphate group, and this PGA is reduced by phosphoglycerol dehydrogenase to yield phosphoglyceraldehyde, and NADPH donates the hydrogen atoms, and energy-rich phosphate group is released. 2 molecules of phosphoglycerol phosphate are used to form starch. So these are the important points during the reduction. Then next, regeneration. During the regeneration of the CO2 acceptor molecule, RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps requires 1 ATP for phosphorylation to form RUBP. So glyceraldehyde 3-phosphate and a DHAP, dihydroxyhistone phosphate, or isomers, and interconversions is catalyzed by triose phosphate isomerase. These are the points, but actually what I explained is here energy for the every 2 carbon for every CO2 molecule entering into the Kelvin cycle. How many ATP and NADPH molecules are required? That means energy calculations for synthesis of 1 glucose molecule. Here, glucose formula is C6H12O6. That means by taking part of 1 CO2 molecule, if starch, that means here 1 CO2, if it takes part in a Kelvin cycle. So to prepare glucose, that should run, our Kelvin cycle should take place 6 times for preparing 1 glucose molecules, because C6H12O6, 6 carbon molecules that should be synthesized. So for the preparation of 1 glucose molecule, Kelvin's cycle should take place 6 times. And for each rotation, here 3 in this cycles, 3 carbon dioxide molecules are taking part and participating. So for this, how many ATP molecules are utilized? See here, total number of ATP molecules. Nine. 6 plus 3, 9. That means for 3 molecules of carbon dioxide, it is needed 9 ATP molecules. That means for the preparation of 1 glucose, this cycle should run 2 times. That means 9 into 2 is equal to 18 ATPs. That is shown here in this table. See here, 18 ATP, ATP molecules are utilized, which turns into ADP. The next, for 3 molecules of carbon dioxide, it requires 6 NADPH. So for 2 times, or for the rotation of 2 times, it requires 6 into 2, 12 NADPH. 12 NADPH are required, which turns into NADP. So currency for this Kelvin cycle are utilized for 18 ATPs and 12 NADPH. So this is about the Kelvin cycle, in which this assimilatory power or energy which is synthesized during the light phase are utilized in this cycle. These are ATP and NADPH. In this Kelvin cycle, again, in this Kelvin cycle, the main important steps are 3. Those are first one is carboxylation, and second is reduction, and third is regeneration. During this carboxylation or this Kelvin cycle, scientist Kelvin first launched this cycle and its changes by using C14 or radioactive carbon C14, and this Kelvin cycle is also known as biosynthetic pathway. Why? Because it utilizes ATP and reducing power, which are synthesized during the light phase in this process, and the end product of this Kelvin cycle is sugar synthesis. And during this Kelvin cycle, the first formed product is a 3C compound. That's why the cycle is also called as C3 cycle. And initially, hypothesis was CO2 is accepted by a 2C compound. That's why it resulted in a 3C compound. But anticipation was there. That is later on they hypothesized that CO2 is accepted by a 5C compound, which results in 6C compound, and which is RUBP, and RUBP is a ketone sugar, and 5C compound. This next to first hypothesis, which was anticipated, and then next they proposed the CO2 is accepted by a 5C compound, which is called as RUBP, and RUBP is a ketone sugar. And here, important enzyme is rubisco, which helps in combining carbon dioxide with the RUBP. And this rubisco is a ribulose, carboxylase, oxygenase, and it is according to the situation, it plays the role of a carboxylase or oxygenase, and this changing depends on the external factors like carbon dioxide, oxygen, and the temperature. Here, once again, that cycle, 3 stages are there. First one is carboxylation, second is reduction, and the third one is regeneration of RUBP.